Genetically Modified Crops and the World Around Us by Amar Dillon, Ryan Smith, Andy Markinsick, and Nick Latham. Our research question is, how do genetically modified crops affect us as well as the environment?
So the main problem we came across while looking at this research question was that genetically modified foods are decreasing the world's biodiversity, thus leaving it more susceptible to ecologic disasters. First, let's start off with what is biodiversity and what does it do? Biodiversity, as stated by the American Museum of Natural History, is the variety of life on Earth, from everything from the genes of the organisms to very ecosystems themselves. This biodiversity basically ensures that all of the organisms of the same species won't go extinct in a short amount of time. This is because if they all had the same genes, they'd all be affected by the same pathogen in the same way. And if this pathogen was to kill them, they would all die. Next slide, please. Now let's talk about what are GM crops. GM crops, as defined by the World Health Organization, are crops that have been genetically modified or have had genes inserted into them artificially by mankind. Because this, and after these organizations do this, they mass produce these seeds on a massive scale and end up having all of the seeds being the exact same. Because of this, now 90% of our canola, 88% of our corn, 90% of our cotton, and 94% of our soy basically all have identical genes to each other, meaning that they could all be wiped out very quickly in the United States. Next slide, please. Let's look at a research study done by the University of Harvard. Here we see that seeds spread away from the GM grass farm that they studied. Every gray seed is where they found seeds, and every red dot is where the seed actually reproduced. The problem with this reproducing is that red dot will reproduce, and then more seeds will spread out from it and reproduce, and then more seeds will spread out from those and reproduce. Meaning that this whole area will eventually be kind of affected by the same kind of genes and genetic diversity. Next slide, please. Again, let's look at the same study done by Harvard University. Here they have a genetically diverse population of potatoes, where there's very many species and stuff like that. And when the pea infested pathogen comes through, it only affects part of them, and so total economic disaster doesn't take place. But then when we have a genetically identical population like we do in GM crops, we end up seeing that all of the crops died from this pathogen that came through that negatively affected their genes. This could be a huge problem if this got out in our wheat population, which is mostly genetically modified, or our corn population. That's why we believe this really needs to be stopped.
With GMOs causing unwanted and unforeseeable economic harm, we need to be able to test their effects in a controlled environment. One way that we can do this is to use the Biosphere 2, which is a closed ecological environment. The reason that the Biosphere 2, according to the official website for the Biosphere 2, the University of Arizona Biosphere 2 consists of a unique, large-scale experimental apparatus housing 7 model ecosystems, which means that the Biosphere 2 is already fitted with seven ecosystems and natural plants, which means using the Biosphere 2 would be as easy as planting a few plants. The reason why the Biosphere 2 is specifically important is because we will have the greatest control over it, because we can change climate, temperature, many different variables, and it will provide the most complete results. The viability of this is that, according to the official website for the Biosphere 2, the mission of Biosphere 2 is to advance our understanding of the natural and man-made environments through unique experimentation, which means that they would likely allow the tests to go through because they want to see the environment, especially how man has affected it. The pros of using the Biosphere 2 are that first, we have complete control over the experiment. We can change as many variables as we want, giving us the most complete results. The second pro of using the Biosphere 2 is that there's no environmental damage from using the Biosphere 2 because it is fully enclosed. A third pro for using the Biosphere 2 is that it is ready to start quickly. We will be able to start testing quickly. All we have to do is plant the plants. The problems, however, are like other GMO tests. It will take time for the plants themselves to grow. The GMOs will have to grow, and they may take many generations to show an effect on biodiversity. A second con with using the Biosphere 2 is that it may need to be reset when GMOs do enough harm to the plants inside of it, which would cost money and once again make getting results much slower. A third con with using the biosphere 2 is that it doesn't really do anything with current GMOs. There's nothing that it can really do to help with those. A major limitation with using the biosphere 2 is that it's only one facility. As good as it is, even though it is the best facility on the planet to test GMOs, there isn't anything more. It's just the biosphere 2. In summary, the biosphere 2 is the best facility to test GMOs, but it is only one facility.
Next slide, please. So, our second solution is to make GM crops infertile. Making genetically modified crops infertile means bioengineering our GM seeds to not be able to reproduce within themselves or in other plant species. This will help decrease the taking over of indigenous species' ecological niches. Why is solution 2 important? As we know, seeds spread very easily and quickly due to sheer number and evolution. Being made dominant resilient species, more and more genetically modified crops survive and thrive, while the local natural ones are being slowly overtaken by the new and improved competition. Next slide, please. Anne Marenborg has a PhD in agriculture engineering and scientific studies at the Norwegian Institute of Gene Ecology. Their research supports that GM crops are potentially dangerous for ecosystems, and they suggest the possibility of making GM crops unable to reproduce with themselves and other plant species. The findings of their studies show that we know too little about GM crops we have created to know the effects on genetic diversity and the natural interactions of ecosystems. Stopping the spread of wild GM crops is a feasible and once researched doable thing, and it's crucial to maintaining the future economy. Next slide, please. Ecologists with the University of Arkansas found a weedy, genetically modified crop called the yellow canola, growing in the wild far from any controlled greenhouse, field, or lab. They decided to request for further research to be done on the unplanned spreading of the weedy canola, and they found that transgenic plants growing in the middle of nowhere, far from fields, far from the controlled biofields, they said. And they also found the same species created hybrids found all the way from China to Japan. These findings were presented at the Ecological Society of America. Next slide, please. So a major strength of this solution is that it would help greatly the environment keep stable and biodiversity balanced by keeping the unindigenous species out of the local populations. We will still have strong, long-living, disease-resistant crops and have unharmed biodiversity at the same time if we implement this solution. But the GMCs already out in the wild are impossible to contain because no matter what, there are just so many that we would have to wait for the old ones to die out, even while our new and fertile ones were living. Another huge con would be very hard to make batch after batch of new crops.
In conclusion, our group feels that solution 1, which is to utilize the biosphere 2, is the more effective solution. We came to this conclusion for two main reasons. The first reason is that using closed ecological environments have been proven to work. A great example of this is the BIOS 3 experiment conducted at the Institute of Biophysics in Krasnoyarsk, Russia. And as shown on the far left depicts a portion of that experiment. The second reason is that using the biosphere 2 would take significantly less time than producing enough non-fertile genetically modified crops to balance out the world's biodiversity. Solution 2 may take decades, while Solution 1 will take a mere months to implement and approximately 5 years to get accurate results. As shown, the graph shown here depicts the use of genetically modified crops from 1997 to 2012. And as Ryan told us before, the use of genetically modified soybeans are very high. And in 2012, reached approximately 95% when it comes to the percentage of planted acres, meaning that on the average farm that uses genetically modified crops, approximately 95% of it grew GM soybeans. Also, Solution 1 will be easier to implement as the resources are already available at the Biosphere 2. Some of these resources include various mechanical devices that regulate temperature and provide rain, as well as soil bed reactors that include microbes that clean the air, as told by the Biosphere 2 official website. Next slide. The works cited page.